All right, so now we're going to cover the cell cycle. And so we have trillions of cells within our body. Now, in order for us to replace cells that are either old, are damaged, or are just dead, what we have to go through is cell division. And so with cell division, this is where we have cells that are going to divide and produce identical cells. And so these are going to be called daughter cells. And so this occurs in two different processes. We have first in this interphase, which is the majority of which our cells spend their time in. And then the second phase that our cells are in is the mitotic phase. So this is when we're duplicating and making these daughter cells. So we'll talk a little bit in depth about this now. All right, so let's start with interphase. So this is this portion that we are going to cover. Interphase, this is where the cell is going to spend the majority of its time in. So during interphase here, what's happening is we have metabolic processes and activities that are occurring so that business as usual is being conducted with our cells, and all in preparation then for that next stage that the cells will undergo, which is going to be that mitotic phase. Now there are three different stages that we have in interphase. We have this G1 or growth 1 phase, we have an S phase which stands for synthesis, and then we have a G2 phase which stands for growth. Now, going back to G1 here, what this is for is the cell starts to grow and it has replication of organelles, we'll have different production of proteins and replication of these proteins, and then also we have the centrioles that are going to be produced just prior to cell division. All right, so then next we have the S phase again for synthesis. So this is where the DNA is starting to be replicated in preparation for then cell division. Lastly, in interphase, we have G2, which again stands for growth. And so what we have here is that the centriole replication is going to be complete. Other organelle production is going to continue on because, again, our cells are a living thing, so it's going to live and die. And then also we'll have enzymes, though, that are going to be produced and that are needed then for cell division to occur when we get into the next phase. All right, so then the second stage that our cells go through is through this mitotic or the M phase. This is where we'll have the cell division, and so this is important for growth and for survival of our cells. What happens here is that we have one cell and it creates two identical daughter cells that are going to be original then to this, what's called the mother cell. Okay, so what's happening here is that we have two different events occurring. We have mitosis, which is going to be the division of the nucleus. And so in this picture here, what we have is the nucleus. And then also cytokinesis, which is going to be the division of the cytoplasm. And if you remember, the cytoplasm has all our different organelles. All right, so just to introduce, there's going to be 4 different stages that we have with mitosis. And so what we have is prophase, metaphase, anaphase, and telophase, and then followed with cytokinesis. And so a great acronym to remember the stages of mitosis is PMAT for prophase, metaphase, anaphase, and telophase. All right, so the first stage of mitosis, we're looking at prophase. All right, so prophase is going to be the start of mitosis. Here what's happening is that chromatin is going to become supercoiled into short, dense chromosomes, and here is the image here. And this is important because what we're getting then is an increase in surface area in a small volume. All right. Also, what's happening is now we're starting to duplicate and make these identical sister chromatids that are going to be joined together at this region called the centromere. And so in this picture, it's going to be that little yellow oval shape that's going to be the centromere. And then these blue lines are going to be then the chromatin. And what's going to happen then, we'll have elongated microtubules that are going to be called spindle fibers begin to grow at each centriole. All right, and so at the centriole then, these are going to start the lining up of the sister chromatids. And once we end prophase, this occurs when there is no more nuclear envelope around the nucleus, all right? And so in this image down here, what we have is the nuclear envelope intact here, but here we're starting to get dissolving of that nuclear envelope, all right? So this will continue on until we no longer have all of that. After prophase, what we have is metaphase. Here, during metaphase, what we have is the chromosomes are going to line up along this equatorial plate, which is going to be the center of the cell. And we'll also have these spindle fibers that are going to now attach all the way to the centromere of the sister chromatid. And so this is going to form this oval structure now, and this oval structure then is called a mitotic spindle. And so here is an actual image now of the cell, and here we have the spindle fibers that are attaching, and here is our equatorial plate with the sister chromatids lining up there. After metaphase, now is anaphase. Here what we have are the spindle fibers are going to pull the sister chromatids apart. So that is right here. And these are going to now pull those sister chromatids at the opposite end of the dividing cell. And this is also called the cell pole. All right. And so now each chromatid is a single chromosome. And now they're going to migrate and separate from that centromere and are being pulled apart so we can start the replication, making that identical cell occur. All right. And so here, if we look at an actual image that's occurring within the cell, so here we have those different chromatids now outlined here that are being pulled apart and that are going to move towards the, in this picture, left and right side to make the two different cells. And the last stage of mitosis is going to be telophase. And so here we'll have the nuclear envelope that's going to form around each new set of chromosomes. And when we look in this picture, here we can see that nuclear envelope reforming, and then it's going to start pinching off, all right? And so the chromosomes now are going to start to uncoil because they have more room to spread out. And now the mitotic spindles also are going to disappear because we don't need the cells or the chromatids to be pulled in the opposite directions. Once we have this cleavage furrow, where in this picture we see it developing right here, this is going to stop the telophase, and what we'll have is then the completion of the cytoplasm. So now we have two cells that are going to be, again, genetically identical to that original cell, to that mother cell, and we'll have the cytoplasm that is going to also form around all of the nucleus and the different organelles. And so the big point here now is that these two daughter cells are going to enter interphase of their life cycle, and then the process will continue on. So a normal process that occurs within our cells is aging. And so this is going to continue on from the day that we are conceived until the day that we die. And so our cells are going to typically decrease in metabolic function as we continue to age. And also they have a decreased ability to have homeostasis, which means regulating its internal environment. And so what we'll also have though with aging is a decrease in number of functioning cells and have an abnormal function that's going to start increasing. And so this is typically, like, as we age, we see elders having more cases of cancers and tumors compared to those that are younger in age. So there's two main ways that cells can die. So at the top image that we have here, this is an image of necrosis, which is going to be irreversible damage that's due to harmful agents or like mechanical damage. And so one example is, and a good one, is when we have a lack of proper care to a wound site. So what's happening is that there is going to be a cascade of events that's going to continue to decrease the amount of cells that can thrive. And typically this is due to a lack in blood supply to an area. And so this is irreversible. Once this happens, this cannot come back. And so if you want that area to go back to normal or pseudo-normal functioning, then you have to have surgical grafts if it's in the skin, or have an an artificial organ that's going to replace it, or have a donor. So the second way that cells die is through apoptosis, and this is programmed cell death. This occurs from the second that we're born. Best example of this is going to be within the spinal cord and for motor neurons. So motor neurons are going to basically compete for survival, and so the ones that are weak and small are going to die off. And so this is a normal event, and it's a use it or lose it principle too. So for muscle cells, especially as we get older, the less that you use your skeletal muscle in terms of like exercising or just normal day-to-day activity, then they're going to die off. And so this is through apoptosis.